Where we are here with carcinogenesis is finally, well, the inactivation of tumor suppressor gene, and now give you a table that we will talk further about what this means to you. So tumor suppressor genes, what are these? They encode proteins that regulate and suppress the two that we already talked about, include RB and p53. Both of these tumor suppressor gene, so that you prevent the cell and we focused upon getting through G1 to and to S phase. Here p53 prevents two things and has two functions. Number one, if the cell can be prepared, it can be. You can recruit your DNA repair genes and then p53 will do that. Repair the cell and put it back in the cell cycle. If this is not possible, then p53 will then recruit those enzymes necessary to bring about apoptosis, so it will guard the cell properly. RB, can you picture RB for me? The RB is bound to E2F. Here, it literally guards the arrest points between G1 and S phase. With the help of who? E2F. What is the name of the enzyme that causes phosphorylation of RB? That is a kinase. Which one do we focus upon specifically? Cyclin D, CDK4. All of this should speak volumes to you when I'm reading this. In activation of tumor suppressor gene, there is something called the two-hit hypothesis. Now this is not a big deal, but well, nothing is a big deal. Once you are actually knowledgeable of what's going on, so let's bring you knowledge. Two-hit hypothesis. You want to think about the mother and the father in order for the patient to then to or progeny to develop a cancer. Then both of the alleles have to be hit. Once they have been hit in the progeny, unfortunately the child is going to or the progeny will develop whatever type of cancer. That is a serial between nodosomes two-hit hypothesis. Both copies of gene must be inactivated for oncogenesis is the theory behind nodosomes two-hit hypothesis. What if you were to inherit one? If you were to inherit what? A mutation. If you inherit one mutation, how many more mutations you need to hit so that you can then develop your cancer? One more. The first hit is that if it's already inherited, then the other hit takes place somewhere down the line. Well, this is then called your familiar. Whereas say that you have a second hit. Now what this means is that no hit. What hit mean? A mutation. No mutation was inherited. One hit in mutation took place sporadically, and a second hit took place sporadically. And this is then referred to as being your acquired or sporadic type of mutation. It is important that you know the term familiar versus sporadic. Lot of times you will find your cancer either behaving in a familiar manner or behaving in a sporadic manner. It is important that you understand which one is inherited and which one will be sporadic. Once again, one that has been inherited is then called your familial. All you need is one more hit to then bring about your oncogenesis, whereas if it is sporadic, you need to have hits taking place somewhere in your progeny. Let's talk about the examples of inherited or familial type of hits. So this would mean that you only require one more mutation somewhere in the lineage of the family tree to develop the cancer. We have familial retinoblastoma. One hit already took place, inherited. A second hit will take place. Now take a child. You do a fundoscopic examination. What are you going to see in the eye? Local choroid. What color is the tumor in the retina? Is this child? Retinoblastoma. White. The prefix leuco means what? White. Leukochorial. Upon fundoscopic examination, think about what a normal retinal examination should look like. Should never look white. So germline mutation of RB, you must memorize chromosome 13. Another very important familial type of germline mutation would be, here is a patient and the patient is losing weight dramatically. He coughing out blood. In addition, there is going to be early satiety. In addition, there is also going to be infertility. There are a lot of issues in this patient. Now you start doing investigation and you end up finding increased levels of the following. Maybe there is going to be the left supraclavicular lymph node that is being affected, and you find the patient BE area negative. That is your gastric adenocarcinoma. I said there was hemoptysis and early satiety. I will talk about referring to your primary gastric adenocarcinoma. I said area negative because it doesn't want to adhere it. Spreads up into left superior clavicular. It is called your virtuous node. Next, I said that the patient was coughing out blood, and here you end up finding upon histology, where chest X-ray shows you one primary nodule, and it is by the mediastinum. One primary nodule. I will just leave it at that right now. And I told you about infertility and such. And then here you might be thinking about on a cut section of the testicle, you might find a non-hemorrhagic type of seminoma in this male. This is interesting. Same patient, doctor. Same patient? Yeah. And you said primary gastric adenocarcinoma, correct? You said one nodule in the lung by the mediastinum, one nodule. Yes, that is the primary. So you have two primary cancers in the same patient. Yes, and just to make sure we are clear, I have given you a third one with the seminoma and non-hemorrhagic. Same patient, yeah, all primary cancers, yeah. What is this? Li-Fraumeni syndrome, a familial mutation in chromosome 17, which knocks out p53, giving you multiple primary cancers in your patient. My topic is familial type of oncogenesis, where one hit already took place, inherited. A second hit then results in major types of cancers. Li-Fraumeni familial retinoblastoma.